Hello everyone. My name is Anuja Charles and I am a biotech student from first year. My topic for the presentation is wearable biosensors in microbiology. So let's discuss about wearable biosensors that are related to microbiology, especially in environmental related field, but yes, it is also discussed and elaborated in clinical fields as well in my presentation. Now, what is a biosensor? A biosensor is a device that converts a biological response into a measurable signal. Now, let it be any biological response which is it carrying out. The biosensor actually converts it into a measurable unit and displays it so that we can understand it. For example, now it has three core parts. The first is biorecognition element, the second one is transducer, and the third one is signal processor. Now, for example, just like how our nose works, that is, whenever a fragrance is being smelled by our nose, and then it goes to our brain where it is converted, and a display is formed in front of our mind that lets us know without even seeing what we have actually smelled. The same way, the biorecognition element acts as a nose, which is that the biological component that specifically interacts with the target, and then it sends the signals to the transducer, which is the brain, which converts that biological interaction into physical or chemical signal. And then lastly, for the display is the signal processor, which amplifies the signal and often sends it to a display or an app. Now this is how a basic biosensor works like. Now let's learn about the whole term of my presentation, which is wearable biosensors in microbiology. Wearable meaning that they are worn in a human body or else in any human body, which which is specifically mentioned for microbiology because the devices whose targets or signals are directly related to microbes or microbial activity. For example, the reasons could be detecting pathogens or for sensing microbial metabolites or VOCs. VOCs are actually volatile organic compounds which are produced by a microbiome or a microorganism as a secretion, as we secrete a lot of things like, for example, sweat. If there is an infection, we secrete pus. The same way, the VOCs are something that are secreted by microorganisms. Now, the third reason could be monitoring for a wound infection, which is a purely clinical based reason. And the main reason which we are focusing on is tracking environmental microbial exposure. And the last one is carrying bioreporter organisms to respond to microbial toxins or pollutants, which is also related to environmental related issues. Now, in case of bioreporter, it is a living organism which we carry, and we'll also learn more detailed about it. Now, what samples do these use? Now, in this case, I have listed only 5, and in which 4 of it is related to clinical, that is related to the human body and how health of the human body. For example, the first one is wound exudate, in which the wound fluids are tested for pH or proteases or bacterial toxins presence, or else the pH difference which is being noticed, so that we could understand whether a microbial activity is being performed or not. In case of sweat or skin secretions, VOCs are being detected, or else metabolites of microbes are being detected by the skin, from the skin or the sweat of the human body. For example, we use sweat sensors or microfluidic patches which are attached to the human body, which detect these. And in case of breath or exhaled particles, this is something which is actually done only by masks, because mask by covering our mouth and nose. Uh, ma- Nose, actually it takes in all the particles which coming out of a human body, and it could capture it and then could be sent to a rapid detection test, and that way we can find out whether a person is infected by something or not. Now, in case of tears or ocular fluids, this is something which is not as common as possible, but the ocular infections which could be found out can be found out by this. Now, this thing which is related to environment is the surface swabs and patches. These are things that are worn, and the sample air or the surfaces which that human body is surrounded with are being in surveillance in case of these. Now, let's see some of the mechanisms that these biosensors use. There are varied types of mechanisms in which I have enlisted almost 6 of it. We'll be detailing each one of it in basic terms. So the first thing is electrochemical sensors. Now, what is, as the term says, electrochemical? It has something to relate with electricity and chemical. Now, what is it? Electro, because it measures current or voltage changes. And why is it, why the term chemical? Because when a microorganism or an analyte is in a body, it reacts, or let it say in an environment which we are basically talking about, it reacts with certain things, and the reactions have oxidation and reduction happening, or else any immobilized receptor could be detected by these through the chemical reaction. Now, this is measured due to the current or voltage change in the chemical reaction. That is the reason it is electrochemical sensors. Now, this is widely used in the clinical range of bandages and sweat sensors. The bandages actually attached to the human body to find out if there is a wound infection could detect is actually detecting that the wound infection is being caused because of the electrochemical mechanism that it is following. Now, the second one, which is actually quite impressive to me, was the optical or the colorimetric sensors. Now, as a first year student, in my second term, I have a lot of colorimetric practicals which are happening, and I do see a lot of color changes which could be measured through colorimeter. So this is something which is quite relatable to me. Now, in case of this sensor, the sensor directly shows a visible color change when it detects something, just as how a pH indicator or a chromogenic hydrogel is used. The same mechanism is used in here, and it, of course, the minute pH changes, or else the minute microbial activity that is being detected is being shown by change of color, and the varied range of the color, that is the varied shade of the color, could also say the amount of how much the activity is going on. And this one is actually the easiest and the cheapest out of all of it, and it is also used in smart dressings, which is also a developing area in hospitals, called as theranostic dressings. Now, in case of the third one, which is immunosensing antibody based, it captures specific pathogens or antigens, and it is all basically used for mass, and it is sent to rapid pathogen detection. Now, the fourth one is nucleic acid based sensors, which is actually a combination of assays which we follow, which could be condensed into a small biosensor that could be attached to your body. Now, this is not a actively participated area, but it is developing. It is only in the initial stage. But what it does is, it detects the RNA or DNA, that is the nucleic acid bases. That is what the biosensors detect of the pathogen. And Especially it is useful in case of viruses. Now how does it do it? The mechanism is as simple as isothermal assays or the CRISPR or the LAMP assay, uh, which is actually a wide process. It requires a wide environment which is being condensed to a biosensor. In case of volatile organic compounds, which is VOC sensors, the arrays detect complex VOC signatures, which is associated to an infection or dysbiosis. Now, in case of dysbiosis is actually any change or any shift in microbial activity. It could be hiring of the microbial activity or else lowering of the microbes, especially in case of a gut. Dysbiosis is actually seen, and that is being detected by these electronic noses. In the lastly, bioreporters. Bioreporters are actually living microbes which are being engineered only for the sake of detecting the signal, only for the sake of detecting the signal, and which is produced by the analyte or the target. Now, this is as the microbe in the engineered microbe is being exposed to the analyte, it shows a certain fluorescence or a pigmentation in itself. That is how it shows it. For example, we have Pseudomonas fluorescens, which is HK44. Pseudomonas fluorescens HK44, which actually fluorescence when it is seen in case of another target. Or else we can also engineer E. coli to be a bioreporter in case, to help in case of this. Now this also has to face a lot of biosafety regulatory hurdles, because of course we have to carry a living microorganism in our body, like it should be wearable, and yet it shouldn't harm the human body itself. Now, in case of applications in microbiology, first we'll look into the clinical and then we'll move on to the environmental based in detail. The first one is infection detection. Now, how does this infection detection happens? The main thing is smart bandages that are used, and smart bandages in such a way that it could measure the pH, temperature, or the protease activity, or else the bacterial toxins, to give early warning that there could be an infection that is possibly to occur. And the recent advantage about this is that from 2023 to 2025, whatever the experiments or the research has been occurred, it has been improved that the smart bandages can also both detect and actively treat the infection by releasing a certain amount of antimicrobial release, or else the giving an electrically assisted healing. Now, in case of the second thing, which is related to the public health, is the mask biosensors. Now, the MIT, Harvard have prototypes for SARS-CoV-2. SARS-CoV-2 is actually the COVID pandemic which was happened the second time, the COVID virus 2. Now, these prototypes were actually made for that, which captures the exhaled virus from a human body, and it reports the infection. Now, this report is actually a government based, or else it should be recorded in some area, like how worldwide there is WHO. The similarly in a state or a country wide, it should be connected so that the report could be submitted. Now, in case of sports or personal health, the sweat or the skin microbiome is being monitored for VOCs, or else for any metabolites to be detected, so that the hygiene or any case of any inflammation that has been occurred does not cause severe issues is being noticed in case of sports and personal health. Now, we'll look into hospitals. Hospitals have theranostic dressings, which is actually smart dressings, which have itself like dressings that detect infection and respond. This is actually an advanced version of smart bandages, but it is used in a hospital based area. in a surrounding which is hospital based. Now we'll come into environmental microbial monitoring. Now in case of environmental microbial monitoring, there are two types as an overview. The first one is environmental exposure badges, in which wearables for healthcare workers or lab staff are being used that sample and signal airborne microbial spore or pathogen exposure in real time. And in case of field biosurveillance agriculture, what is that? It is the wearables used by field workers to monitor the exposure to plant pathogens or else fungal spores or contaminated water while on the job. Now this is actually, even though the workers are being using this is not actually aiding the host as of directly, but it is used to detect from the environment rather than from the host. This is why it is environmental microbial monitoring. Now to detail about it, wearable biosensors that can be adapted for environmental microbial monitoring are actually used to detect the microbial contamination in air, water and soil. Now, they can monitor pathogenic bacteria and bioaerosols in polluted environments. It helps in early warning of environmental health risks, and it is useful in industrial zones, waste treatment sites and water bodies. Now, it is used as a real-time detection of microbial pollutants. So, it helps monitor the water quality by detecting microbial toxins. It detects the microbial activity in sewage and waste systems. It supports the environmental protection and public safety, and it promotes the sustainable and preventive environmental monitoring. Now, these are the main uses of why do we use the environmental monitoring through a biosensor. Now we'll look into some examples of the environment related biosensors those are used. Now the first one is wearable bioaerosol sensors. It is used to detect the airborne microorganisms. It is mainly used by the environmental workers in polluted or industrial crowds, and it helps in monitoring the microbial air quality. Now, in case of wearable VOC, which is volatile organic compound biosensors, it detects microbial metabolic gases. Now, as it is gases, it is seen that it could be have been used as an air quality itself, but to be frank, it is used in wastewater treatment plants, landfills and sewage systems, in which yes it does determine the environment, but where the pollutant or the analyte is actually present in a fluid quantity is moreover being looked after. Now these microbes release VOCs during decomposition. Now the third one is wearable water quality biosensor badges. Now it detects E. coli and coliform bacteria indicators. Its use is that by field researchers and environmental inspectors who inspect the water quality of a river or a pond or anything and have to survey them out. Now it gives real warning of microbial water pollution. The next one is smart environmental monitoring variables, which is used in agriculture and soil microbiology to detect the activity affecting soil health, the activity of microbes affecting the soil health. Now, in case of the last one, which is wearable biosensors for waste management workers, it detects the microbial load and toxins and reduce exposure to harmful microorganisms, and it is applied in environmental safety. Now, we'll move on to key engineering and microbiology challenges. The first one is the sensitivity and selectivity that it causes. Now, the microbes are often present at low concentrations. So, distinguishing it with a pathogen or else distinguishing it with a normal human body which has different could have variations in the pressure or the pH is completely not possible at all. That is the reason the sensitivity is that whenever by chance there could be false positives and everything, false detection of a microorganism which is not present in your body, but it only happened because of the sensitivity of the biosensor. Now, in case the interference from environmental noise and pollutants. Now, this is totally related to the environment based, which is actually uh, the environmental pollutants are of varied kind, and we cannot segregate it in case of the air or water or sewage. Now, the mixture of all of the pollutants of different kinds can also disturb the biosensor. Low microbial concentration in environmental samples. Now, this is because in some cases the biosensor is expecting a certain amount or a certain level of pollutants which is not achieved, but doesn't mean that the air quality is pure or the water quality is pure. It is just that the biosensor is expecting a little higher than it is actually. Doesn't mean that the air quality could be verified as it is correct or a normal condition. Now, the third one is biofouling and stability, which is actually just because this is a wearable biosensor which we are discussing about. The sensor is used for long term, and it can cause degradation or signal drift. It could either cause degradation for itself, or else it can cause harm to the host, which is the human body which is wearing it. Now, in case of false positives and negatives, this is actually based on clinical. Now, the stakes are quite high for infection detection. So, the devices must be validated before extensive use of it. In case of power and data, it is quite easy and reachable because it requires low power, it has wireless transmission, and it secures data handling. Now, in case of regulatory and biosafety, devices that detect pathogens and also the engineered microbes, which is actually the bioreporters that is mentioned, face stricter approvals because you're being treated with a microorganism. Your sensor is reacting with a microorganism or is containing a microorganism which could be harmful to a human body, because of course microorganisms in especially pathogenic are considered severe, and detecting and storing it for further tests, or else carrying it to detect other microorganisms is a hazard. The last one is user compliance and comfort, because the as it is wearables, it should be comfortable and unobtrusive in nature to the human or the person who is wearing. In case of safety, ethics, and regulatory issues, the main reasons which this came up is because yes, the first reason is as it is wearable, it can cause skin irritations and allergic reactions. Now, these are few concerns raised, making the reason for the regulatory issues that are happening right now. So, the first one is skin irritation, which is a concern. The second concern would be handling of microbial samples. And the third is privacy concerns, like the data which is handled should be secure properly. The fourth one is need for the regulatory approvals, which is actually the format or the procedure that should be followed, which is quite long for any biotech related field for now, or else especially biosensors, because it's a developing field and it has not been recognized, or it has not proven its point yet. The last one is biosafety levels. Yes, the levels of biosafety should be considered and it should be passed. Now, this is related to the biosensors presentation which I have prepared about. And thank you so much for being so patient. I believe that you would have been enriched with the data, and this data is useful to you. Have a good day ahead. Thank you.